Breast cancer is the most common cancer in women worldwide, and metastasis is the primary cause of death in patients with breast cancer. Cannabinoids are well known for their diverse medicinal properties, including anti-inflammatory and anti-cancer effects. Cannabidiolic acid, or CBDA, is a minor phytocannabinoid found in hemp fiber and seed oil that is often overlooked and receives less attention than, for example, its neutron analog CBD. However, CBDA has been demonstrated to be an active compound exhibiting pharmaceutical features similar to other active cannabinoids. Particularly, CBDA has been reported to have anti-cancer properties against highly aggressive breast cancer cells. Therefore, this study aimed to investigate the potential of CBDA in inhibiting the metastatic progression of MDA-MB231 breast cancer cells. Thus, it was hypothesized that CBDA would hinder cell migration and modulate genes involved in promoting and suppressing metastases. This study was conducted in two main sections. First, I treated the cells with CBDA at 20 micrograms per ml with 24 hours of incubation time. Then, I analyzed cell migration using two migration assays: horizontal migration with the scratch assay and vertical migration with Boyden chamber assay. Second, I studied gene expression after the same treatment conditions using RT-qPCR, analyzing COX-1 and COX-2 expression, expecting to have a downregulation of the gene as a positive regulator of metastasis, and SHARP1 expression, expected to have an upregulation of the gene as a suppressor of metastasis. The results show an arrest in cell migration of the breast cancer cells with CBDA treatment, as is illustrated here in the representative pictures of the horizontal assay, in which the scratch remains after 24 hours of treatment, in contrast with the vehicle group control, where some level of migration is observed after 24 hours treatment. This resulted that CBDA significantly inhibits the migration of this highly invasive breast cancer cell line, as it exhibits a lower wound fill percentage post-treatment. These findings are consistent with previous studies, which indicated that CBDA at even lower concentrations inhibits cell migration. On the other hand, vertical migration assay, the outcomes reveal no significant decrease in the number of cells migrated. This can be suggested that these conditions were insufficient to see observable results of migration arrest in this particular assay. Next, looking at the gene expression evaluation, we can observe a significant reduction in the expression of COX-1 gene, around 0.49-fold change, and a significant increase in the expression of SHARP1, around 3-fold change, in contrast with the control groups. However, contrary to the expected, CBDA significantly increased COX-2 expression around 5.2-fold change, contrasting with previous experiments in which CBDA shows anti-cancer effect by inhibiting COX-2 enzyme and downregulating its expression. In conclusion, CBDA inhibits the metastasis of MDA-MB231 cells by arresting cell migration and regulating genes that promote and suppress metastasis. These findings present an exciting opportunity in using CBDA as a potential future therapeutic agent for treating breast cancer, and also CBDA can be better employed from the hemp industry byproducts. Further studies can be conducted to compare the effects of CBDA using different cancer cell lines or against FDA-approved anti-cancer drugs. Moreover, additionally, research is needed to understand the molecular mechanism of the inhibition effect, or in combination with other cannabinoids to identify different effects.